Good morning, everyone. Thank you for your patience as we get started here this morning. I want to welcome everyone to the Bell webinar series for 2023. We're excited to share new research and ideas in the neuroscientific community. Today's webinar is language acquisition using HD EEG. Our presenter is Dr. Ghislaine Dehaene-Lambertz, and she has pioneered the use of brain imaging for infants, and her talk will focus on the study of early language acquisition. Dr. Dehaene-Lambertz is the director of the Developmental Neuroimaging Laboratory at Neurospin, INSERM, CEA, Université Paris-Saclay. For questions and answers, we would ask that you write your questions into the chat feature of Zoom, and we will answer them during the Q&A period. So I'll go ahead and turn it over to Dr. Dehaene-Lambertz.
Thank you very much. It's really a pleasure to be here with you, especially to speak about high density EEG recording system, because I think that it's thanks to Don and AGI first and now Bell that I has been able to do all this research that I want to present to you today. Okay, so the idea today is to show you how we can use high density EEG to explore language acquisition in infants. Okay, so just to remember you how the power of speech, because with speech, because of the elementary bricks that are in speech, we can combine them to exchange information between two minds about the world. So this is a very big and crucial capacity that humans have. And to use language, infants should do several stuff. They should be able to process speech at the different levels of a complex hierarchy who go from phoneme until words and sentences. They should discover how their native language has implemented the rules to combine these elements. They should understand that these operations are done to represent, so you have an interface between the linguistic system and semantic knowledge, and to exchange information with others, so you have also an interface with social cognition. So it's quite complex to set up. And I think that words, to study how words are represented, is a good way to look at two different aspects of this system: the interface between the acoustic system and the phonological and linguistic domain, and also to look at how the baby understands that this auditory input correspond to refer to something in the outside world. And I will present these two aspects during this talk. So if you are a baby and you need to learn words, you have several problems to resolve. The first one is that speech is a very variable signal. And so you have to find what is the elementary unit on which you can rely to obtain stable and robust representation. For example, here you have my deep French accent, and still you should be able to, I hope that you are able to recover what I am trying to say, despite the fact that maybe my phonemes, my English phonemes are not very correct. Speech is also a continuous signal. It's not like written language in which you have a blank between each word. Here you have a continuous signal and you have no pause between each word. So how infants can chunk this signal to find putative words. Also, what do words represent to an infant? So if you hear a cat who is doing meow, meow is not representing a cat, but if you heard the word cat, is that also an association between this verbal form and this animal, or a true abstract symbol representing or referring to that animal? And finally, my final question is, can we find early evidence of these processes, or do they develop slowly with the duration of exposure to speech? Given that I am working in infants and that I want to present positive results, you can already imagine that the response to the last question is that indeed we find early evidence of all this process very early on during the first weeks of life and even in newborn. So the first question, how infants can cope with the variability of speech? So as you know, there is no simple and robust relation between the spectrotemporal pattern of the speech signal and the perceived phonemes. For example, and I will show you example of place of articulation is one of the most difficult phoneme or contrast to perceive. And you know that when you are speaking on the phone, you are obliged to say be like Bernard, for example, or de like Denis, because otherwise people do not understand. And when you are a dyslexic kid or when you suffer from stroke, you very easily lost the difference between these two phonemes because they are very close and still they are distinguishable to separate words. And we have, as human, normally, if we have no stroke, and we are not in noisy condition, we are able to categorize these phonemes despite different voice, different accent, different type of production, and despite the fact that vowel or cluster of consonant can modify the spectrogram of B, for example. It's true that animals can be trained to discriminate speech sound, but their success remains generally limited to the speech set they are trained with, and they have difficulty to generalize, for example, to other circumstances. And in fact, they do not possess the many properties that characterize phonetic perception in human adults. So now that I have said that, what about the human infant? So the idea that we have a long time ago is to, because infants are not able to answer to us whether they perceive or not, but so the idea was to use EEG and in experiment in which we can reveal whether they perceive the phonemes with the same property that characterize adult perception of phoneme. So what are these properties? Categorical perception, normalization across voices, and normalization across co-articulation. So our first experiment, and this one was done thanks to Don, because I came to Eugene and it was the beginning of AGI, the beginning of this nice net. And they were only testing adults, and because I am working in babies, I said, haha, can we do that for babies? And it was the first net that we get with 64 electrodes that very kindly done, and other people in maybe it was even not AGI at that time, construct for me. And with this net, we tested repetition of syllables. So we presented to 3 months old series of syllables like ba, 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 or ga, 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 ba. And we were looking to the last syllables of the series, depending on whether these syllables was preceded by the same syllables in the yellow case or by the other syllable. And as you can see, you have a very nice habituation of the response with a decrease of amplitude of the voltage when you repeat the syllables, and when you change the syllables, nothing happened for the first peak. As you can see, you have very nice peaks, 2 peaks here. And the second peaks at around 300 milliseconds, you have a recover of amplitude of the response. And if you look at the topography, the very nice topography that you can obtain with high density net, you see that you have a very significant difference between the standard and the deviant type of striol. So just to give you an idea of how robust are these results, we just finished another experiment here in full-term neonates. And now with much more clever statistic than at that time in 94. And you see again that when you change, it was exactly the same syllables. We tested them again, and you got the same beautiful mismatch response with a positivity on the frontal lobe and a negativity, a reverse of polarity on the back electrodes. But okay, babies and even neonates are able to discriminate syllables. But remember, I said that I want to test different property of phoneme perception in adults. And one of them is to be able to generalize across voices. So here in that experiment is two different syllables, pa and ta, so very close syllable again the place of articulation. But in the first row here, it is the same syllable, the same speaker, and exactly the same syllable, which is repeated, and you go to another syllables. But here, what is interesting is that it's different women who were producing the syllables. So it was pa, 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 ta. And despite the fact that the pa or the ta were produced by different speakers, you still have this mismatch response, showing that the difference between the phonemes was, in that case, still perceived by the neonates, despite the fact that they were produced by different speakers. The second property, which is important to test, is whether phonemes are perceived categorically. So here we have a continuum, a physical continuum between ba and da. So it's just a change of the slope of the formants. It's artificial speech. And so you have exactly the same change between each of these bars. And you present to the baby the same type of trial, so in which you repeat the same syllable. So for example, control here, you repeat ba, 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 ba. Or you come from this green point and you do ba, 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 ba. But you see, you are within the same category bar, even if you have an acoustic difference between the green and the yellow points, because it's speech, you do not perceive it. But if now you are coming from this red point, so it's the same acoustical difference between the red and the yellow points that it is between the green and the yellow points. But now you go across a phonetic boundary and you perceive da, 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 ba. So the question was, will we find a stronger difference when you have when you cross a phonetic boundary relatively to within category change? And it's again 3-month-old infants. Why 3-month-old infants? It's just because they are easier to test. They are very kind, and parents are a little bit after all the tremendous agitation after birth, so they are also calm and quiet too. So it's the best age to test this type of things. So just to look again to the repetition, so you see the habituation of the ERP when you repeat the syllables. But when we change, so when we arrive to the yellow point here in yellow, you see that if you come from the green or the red point, you have an increase of amplitude of the voltage when you have this change. And this increase is much bigger when you are coming from the other side of the phonetic boundaries and when you are within the phonetic category. And when you look at the topography, you see that indeed the across category change induce a much larger and wider response than the within category change, with a significant difference here. So it means that the babies just listening to these syllables display in their neural response categorical perception. So that was already quite old also, 98. Time is going fast. But the following question we had is whether they perceive the consonant despite the co-articulation. So we have seen that they perceive the phonemes categorically, that they are attentive to a small difference between two phonemes when it's phonetically pertinent or relevant, and that they neglect the voices. But here, what you can see on this plot is that when you go, for example, from D to du, the fact that the de is followed by E or U syllable, vowel, it changes the slope of the transition between frequencies. So the spectrogram changed because of co-articulation. So it could maybe change also the perception for babies. So we did exactly always the same type of experiment to represent a series of syllables, but now they share the same first consonant, but the vowel is changing. So the big thing is that in French we have many vowels, so we can do many type of these syllables. So you can have ba, bon, be, bi, and you compare. So that's a match trial despite the change of vowel at each syllable presentation, and you compare at gain, ga, ge, bi. And again, you see that we have this mismatch response with this positivity on the front that is corresponding to a very larger response to the deviant trials relatively to the control trials. So infants seems to perceive the similarity of the consonant even when the adjacent changes. With Karima Mersat that you see here, we wanted to go a little bit further because the idea is that maybe in the acoustical space or acoustical phonetic space, it's not exactly that you extract the consonant, but with the vowels, they are in a similar space of this phonetical space. But maybe babies do not really perceive the consonant as the same. So an idea that we had to show that indeed babies are perceiving the consonant per se and that they are able to manipulate the consonant within the syllables was to train them to read. It's not really to read, but it's the same idea that if when you read what you are doing, you are just associating a phoneme with a visual shape, with a letter, with a grapheme. So we say, okay, we will do the same. We will present to babies syllables, for example, gi. And after that, they will see a yellow plus, as you can see here. So they had a visual a woman in front of them just to keep them attentive to the center of the screen. And they hear one syllable, so gi, and after that, the yellow plus, or if it's be, it's a red bar. So each time they hear a syllable starting with ge, it's a plus, the yellow plus, and if each time we presented a syllable starting with be, it's the red bar. And we trained them with not so many trials, 36 trials, and after that we introduce ba and ga. So they have never been presented with these two syllables, and they, we present it's the trials with ba and ga are 50% of the time associated with the yellow plus and 50% of the time with the red bar. So if they just associate these shapes with syllables, they know nothing about ba and ga. But if they have understood that the shape is associated with the phoneme, the first consonant, they will be surprised when ba, for example, is associated with the yellow plus. So first you can see here the response, the visual response to the shape with the P1, P400, which are the classical visual response in babies when you are looking at the occipital electrodes. So that's for all the trials. And when here you see the response to ba or ga when they are congruent with the learning, you see this big red, green response. But when it's incongruent, you have like a flattening of the response. So a very significant difference between congruent and incongruent trials. And a very long with a slope, slow responses in that case. So it means that babies can extract the consonant from a CV syllables to associate it to a shape. It's not that it can be done spontaneously, I think, because in that design, we attracted their attention to the correct level of analysis. Okay, so to summarize a little bit what I have said till now, with all these experiments, we have found that preverbal infants have in fact the full set of properties which are used to define phonetic perception in adults. They normalize across voices, they have categorical perceptions, they normalize across co-articulation. We have other experiments showing also that they are able to associate the visual shape, the visual perception of the articulatory movement of an adult with the sound that this type of movement should produce. But the questions that I have, remember you on the first slide, what was the code do they use? Do they perceive the whole syllable? Probably not, because I just said that they are able to extract the phonemes from the syllables. But if you look now on phoneme, can they even go to phonetic features? Because you know that phoneme are defined by like a binary system of binary traits, like for example, a place of articulation, manner of articulation, etc. So, what is the unit in which they rely to perceive speech? So that was the second. If I may say, a nice exploitation of Don, because I asked him to build for me a very high density system. The idea at that time was that babies have a very thin skull. So if we place a grid of electrode on the temporal areas, maybe I can have not an EEG measure, but something much better than usual. So Don and Amy, who was working with Don at that time, replaced each electrode of the 128 classical net with this grid with 7 electrodes. So now I have a tight grid of the temporal areas in babies. And with that, and with Julia Guénari, we wanted to decode syllables that were presented to the baby. So we present to them 120 different syllables. Why? Because we take syllables that were contrasted on the place of articulation, be, de, ge, but also manner, so be and me. So we have be, me, de, ne, ge, nye. And we, because we wanted to have the co-articulation, so they were produced with a E or O and produced by a male and a female, and each one of these speakers produced 5 exemplars of each syllable. So many variations. And we presented 1 syllable by second during 1 hour. And to obtain that from infants, they were just asleep. Because otherwise it would have been impossible, but because we wanted to decode their response, we wanted many exemplars, and so we tested them asleep. So here is a beautiful ERP, because when you have a 3,600 trials in one baby, it comes very nicely. So you see the ERP again, it's very reproducible auditory response of infant of that age. But it's not that what is interesting, is to train decoders and to try to see whether these decoders can find a set of parameters to separate this phoneme, and we will see that a big part of this work is to see whether we can generalize across different parameters. So indeed, we were able to decode manner, place, and vowels. And as you can see, it's not exactly at the same time. So you have the performance of the decoder a long time. And when you have this little circles, that means that it's significant. And manner is easily decodable because be and me are very different temporal envelope. So it's quite easy. In fact, it was a security for us whether we would be able to decode manner. It was just to show that the experiment was working. A place of articulation is difficult to decode, as you know, because from what I said, it's something which is very confusable. There is not a clear spectrogram for place, but still we were able to decode place, and vowel, because it's CV syllables, we can decode the E and the O, and it's a little bit later because the vowel arrived later in the signal. So we were already happy, but it was not sufficient, because maybe with all this we can find any parameters and can decode and can separate these syllables, but it's not what the brain uses. What the brain is doing is that it is able to generalize across non-pertinent variations. So here what we did is that, for example, we train the decoder on the male voice and we test them on the female, or reciprocally. And even when you do that, you can decode manner, you can decode place. You can also train on the syllables with the E and test them on the syllables with O. And again, you see, you can generalize quite nicely across the co-articulation produced by the vowel. What is more interesting is now to try to decode across the phonetic feature. So here, what we are doing for manner is that we train on B versus M, and we tested on D versus N. So if I come back to my table here, so you train here B on this part of the place, so the most frontal place of articulation, the more labial place of articulation, and you test on this one or this one. So you are obliged to generalize not only across vowel, voice, etc., but also across the phonetic feature. And so what is interesting is that for the first part of the signal, so during the first 200 milliseconds, you are able to generalize across the other phonetic feature. But in the second part of the signal, now you are no more able to generalize, because now in this second stage, B is no more D, because it's a phoneme which a different combination of phonetic features. So now, here you have in the first step only computation of the phonetic features, and in the second stage you have the computation, the merge of these features to create the phoneme. So to finish this part on the acoustical phonetic perception of babies, speech is processed in parallel by different pathways in the perisylvian areas. That's data that we obtained from MRI and also type of EEG experiment we did. But one of these pathways allows the infant to factorize the speech input into orthogonal vectors, which are the phonetic features described by the linguist, which are subsequently recombined into phonemes. So it is a very interesting way of doing, because if you have such an encoding system where you project the auditory input on a finite set of minimal and orthogonal elements, which is the phonetic feature, you get rid, in fact, of the irrelevant variation, and that provides robust unit for subsequent combination in higher units. So I think that it's a very important results to show that, in fact, babies are able to have this code based on phonetic features. So now that you have the phonemes, what can you do? Because you have a second problem, which is to cope with the continuity of the signal and to chunk the speech stream, because I said it continues. So you have, how you can do that? You have a few words which are produced in isolation. You have also the prosodic cues that can help you to segment the speech stream. And what has also been proposed is that the fact that in words you have you can exploit the fact that a word is constituted by precise syllables and that you have higher transition probability between the syllables constituting the words relatively to the syllables between words, because any words can be followed by any other words, so you have a drop in the so just to give you a little bit of the idea, I hope you can hear the... I don't know if you hear it, but the idea, it was an experiment done by Jerry Safran, Lisa Newport, and Ghislaine in 1996, in which they saw that when you present a stream like that to babies, to 8-month-old babies at that time, in which, for example, lutiva being one of the pseudo words used in this stream, lu is always followed by t is always followed by va. So lutiva is a word, but va is followed by any of the other words, so fobumi, dinago, or zazogu. So you have a drop in the transition probabilities between the syllable va and other syllables. So babies, it was a proposal by the authors, are able to compute this probability and to extract the words. And after that, when you present to them words and part words, they they look longer to the part words. So the question we had with Anna Flo and Benjamin Lucas was whether infants were really segmenting the speech stream during the familiarization. Because in fact, here with behavior, you have no idea of what is happening. You just, after that, present trisyllabic words like dinago versus nagolu. So you give the solution. You say to them, it will be trisyllabic words. And maybe after that, so they have these trisyllabic words and they are more familiar with dinagos than with nagolu, without having really segmenting the words, the stream in words during the learning part. The second question is that if they have segmented the words, how precise is the word segmentation? And from what age can they use this type of strategy? So with Anna Flo, we test neonates and with EEG. That's the big advantage of EEG, is that you have you do not need behavioral response. And the second advantage is that you can test them asleep, which is also interesting. And we present, what we did is that we have a little bit of resting state. After that, we have a random presentation of the syllables. So I don't know if you can listen, it's just the same syllables. But, and after that, we have the same ideas that Jerry Safran, four trisyllabic words which are randomly concatenated. And the idea is that with EEG, you can look at the neural entrainment. That is, if a brain receives syllables, because they have all the same length, syllables every 250 milliseconds, it will begin to follow just because of the speech envelope, and you will have an increase of power at the frequency of the syllable. If now they are able to group the syllable in trisyllabic words, because you have trisyllabic words and a trisyllabic words and a trisyllabic word, you should increase also, in that case, the power at the frequency of the word. So it's what we look at it. So here you have the resting states, and you have the topography at 133 Hertz, which is the frequency of the words, almost 4 Hertz, which is the frequency of the syllables, and during resting states, there is nothing. It's what we were expecting. During the random string, you see that you have many electrodes showing an increase of power at 4 Hertz. And I don't know why this red arrow is here, but when now you present the structure stream, you see that you have indeed still an increase at the syllabic rate here, but you have also an increase of power at the word rate. And when you make the subtraction, you see that you have indeed more power at the word rate in the structure stream, so in the Dinapo, Lutiva, Fubini stream, than when they were listening to a random stream. What is interesting also is that you can follow learning during the stream. And so you have a sliding window. It's why you have this ramp, a little bit of ramping of the effect. But you see in red the increase of power for the syllabic rate. So from the resting state in the random, the learning part. And after that, we have a test part that I will explain later. And for the words in blue, you see that you have an increase during the learning. And in fact, with these babies, with neonates, sleeping neonates, they need 2 minutes to show, to display for us, at least in the EEG signal, an increase of power for the stream. So it means that with this type of technique, we can see online the online computation of the transitional probability, which lead to a chunking of the stream every three syllables after around 2 minutes of exposure in a sleep neonates, which is quite an ability for these young babies. And in fact, we have found, so the statistical learning, this type of response is very easy to obtain in many situations in neonates, and even we got some response also in some comatose patients. So after that, maybe I will go through it because it's too, it's coming, it takes too many time. Do I have still time or not?
Oh yeah, you've got.
Yeah. Because so I will go quite fast for the second part, which is based on symbolic representation and how a baby can use words to represent the world. So, okay. This line of experiment starts with a very nice experiment done by Sandy Waxman, published in PNAS, in which he showed to very young babies, 3-month-old babies, images of dinosaurs. So, for example, babies were seeing that, of a succession of images of dinosaur, and when they were habituated, it was a looking time experiment, they saw this dinosaur and a fish. And so it's quite bizarre. You have 3 months old, they have never seen any of these animals, and what is happening is that indeed they are not able to say, they did not display any specific behavior when they were looking at these images. But in a second experiment, Sandy Waxman said each time there was a dinosaur, say, look, a Thomas, there is a Thomas. So each time you have this dinosaur, you have the word Thomas, which is arriving. And what happens in that case is that when you have presented these images with the word Thomas, the 4-month-old orient significantly more to the new category than, so they seems to have make a category of the dinosaur. So what is happening here is that when you give a word to humans, to human babies, they try to find a common feature between all these animals. And as you can see, they all have legs, a neck, and so on. And they are quite different of this animal, which is in only one whole shape like that. And so we think that it's because babies know that we communicate with speech and that speech convey information, that when they hear this reproducible word, it attracts their attention to something that should go together. And it's working with words, but not at all with backward speech in purple here, nor with tones. In a funny experiment, Waxman shows that if you present to babies a video movie showing adults exchanging with tones. So for example, doing do, 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 and the other one answer do, do. After that, they accept tones to be representative of the category here. So naming the picture helps for Monsol to form the Toma category. So we were interested to do that with object also. So we had now a set of images like birds, and we call them pauvans, and a set of other images that we call kafu. And we present to the baby a little bit like in the reading experiment. So they see eyes to keep them towards the center. They hear the word, and after that, like pauvans, and after that, they see the object. And another group did the reverse. They see the object, and after that, listen to the words. Again, very short trials of training, only 36 trials, and after that we start to present incongruent trials. In that case, you say pauvans, and you get this image. And so, as you can see in the word image group, we have a very nice difference between congruent and incongruent trials. Again, you have they respond less to the incongruent situation. So here you have the incongruent, the congruent, the difference, and the cluster-based permutation analysis showing the statistical cluster, the significant statistical cluster. So you see difference in the occipital areas for the image, and when you look in the reverse, so when you see the image word group, you have a response which is mainly anterior posterior, but more followed by a late response. So you get larger modality specific response for the expected stimulus and a late response in the image word group. So it means that we confirm what Waxman showed, and that babies can learn quite fast to associate a word and a label, and to classify all these objects, these animals in one under the same label, and oppose that to this other label in both directions, coming from the word to the image or the image to the word. I just want to finish to present you this experiment in which we were wondering whether it was just an association or more than that, whether indeed the label, the word can act as a symbol for the category and not just as an association. Why we are thinking that? It's because, in fact, babies seem to very easily learn the pairing between an object and an arbitrary auditory or visual sign. So, for example, they can learn already some first names. They learn the toma. They learn quite fast also to associate the consonant with a red bar, for example, and here these images with this arbitrary word. They do that in a few trials in both directions. And we think that maybe while animals, it's much more difficult for them. And okay, so so what we showed, in fact, it's across several experiments that indeed when you teach in one direction. So, for example, if you present an object and you say the word after, the babies are able to go in the reverse direction. So if I say kafu, now they will wait for bird if they have learned in the direction bird to the word. And we have evidence that animals are not able to do it. I will just jump to that because I think it's more interesting. So we did it the same in adults, and we have it's in MRI and in EEG in babies. And it's just to show you that indeed we present a word followed by an image, or an image followed by a word. And when you do that, adults even don't know that you have reverse pair, and you have the same activation by the reverse pair in blue than by the canonical pair in red. And in fact, you have this green part, which are all the common areas. What is interesting is it's not only the linguistic system, but also on the right side, you have a very symmetrical response. But what was interesting, and I just want to finish on that, is that when you look at macaque, they are not able to do it. First, it was very difficult to teach them the association, and we had also visual association, because indeed the speech is more familiar with for humans than for macaque. And when you make an error of an incongruent trials, they have learned, so they have an error, but when you reverse the pair, they are not able to detect the incongruity, meaning that they are not able to, so they have an associative learning going in a specific direction, but they do not reverse immediately. So I think that the time is too fast. So just to conclude, preverbal children are really able to circumvent signal variability by factoring into orthogonal components, the phonetic features which are subsequently recombined in phonemes. It's what I said with the decoding experiment. They have powerful ability for statistical computation, even asleep. They encode in memory chunk of the signal. They easily learn and use label for categories. And we have shown also that they are able to use these labels in operation like negation. And so I think that really human infants are really a very fast learner, and probably because they have a dedicated neural processes underlying this ability, and it is because we are able to use very now non-invasive brain imaging techniques that we can look at these capacities. So here are the people who have worked with me on that, and I recover these old images showing done with the first net that we used in 93. And the last net that he did for me, and I hope he will do more to record babies. And thank you very much, Don, because I think that it was thanks to you that I was able to do all this research. Thank you.
Wonderful. I think we have time for some questions if people want to put questions into the chat. And it looks like Don has a couple of questions here. In the classifier's features, are there clues to the topography of the EEG that are discriminating, maybe consistent across different phonemes?
In fact, no, because it was very variable. When we look at the electrodes that were we had weight in the decoding classification, it was not the same across all the babies. So it was difficult to find something very reproducible. So I don't know what does it mean, because topography is always with EEG quite difficult to interpret. I wanted to add something, but so the high sampling net, high density net was useful much more for we did an experiment to decode number. It was much more informative for number, but still it was informative. But after that, whether we can attribute a specific topography to one or the other feature, it was not possible.
We have another question. Are you planning to test 6-month-old babies?
I do not have why 6 months old. In fact, I would like to go rather to the young age, because what I am interested in is really what do we have to start with. So how is the human brain at start to do all we have to do? So I am more interested by the younger and preterm. We did some experiment in preterm babies that to go in older babies, and they are moving more, which is also a problem as soon as you want to do brain imaging.
Don has a question. If the brain is decoding sounds while asleep, could we use these responses to test coma patients to determine if their brains are working?
Yes, yes, yes. In fact, there is experiment using the mismatch, for example, to predict coma. How do you say? If you get a MMN response, usually they will go out coma in the weeks that follows. So it seems that you have like prediction of that. We have looked at statistical learning in comatose patients, and what we find is really that the depth of the coma is related to the syllabic response, not so much to the word response. It was difficult for the word, but for the increase of frequency at the word, the increase of power at the word frequency, it was difficult to find a clear time. Was not very clear, although we had some response at that frequency in a group, but not really for each individual. The power at the frequency was much more interesting.
And I just want to thank Dr. Dehaene-Lambertz for that wonderful webinar, and I want to thank everyone for joining us today. And if you missed part of the webinar, we will be able to put it up online.
It was a great talk, Ghislaine. Thanks very much.
Thanks, Ghislaine.